parental generation. So those were the first parental generation, and they are usually represented by P1 generation. Then the first offspring that are produced, they are known as first philo generation. All right, that is what, and they can be presented as F1. All right, so we've got P1 and F1. All right, so these, all right, so these are called what? These are called parental first parental generation, and these offspring are known as the F1, which as the first philo generation or offsprings. So now what I'm going to do here, I'm going to use the first generation, and I'll do the experiment again and see what sort of offspring will these parents give us. In with our second experiment on genetics. So remember, in our first we had our F1. So now in this I'm going to use those F1 parents, all right, to do the experiment. So again, we'll do the same thing. Let capital letter T be the allele for tallness, all right, for tall, and capital letter T to be for allele for short. All right, that's what we're going to do. Then second thing, now we have to come up with the parent genotype. All right, so what will be the parent genotype? All right, so the parent genotype, the parent phenotype, that is the first thing, the phenotype first. So the phenotype will be what? Will be tall, and even this time we've got tall, because remember those offspring were all tall. Then what will be the parent genotype? So the parent genotype, in this case, all right, it was like this, isn't it? So that is the parent genotype. Then let's have the gametes. What will be the gametes? So the gamete, we're going to have tall, all right, and we're going to have an allele for the short, but just remember that this is dominant, all right. And here again we'll have the same thing, and these are our gametes. Then let the experiment begin. All right, so this will cross with this. What will be our offspring? All right, so our offspring will be all right, we'll have that. And again, the same is, we'll do the crossing with that, and we'll have capital letter T and small t pairing up like that. Then we'll come this side, this will do the, all right. Then what are we going to have? Again, we'll have capital letter T, so it's always to start with the capital letter, yeah, to show respect. That is what we have. Then last but not the least, again, this one will cross with that one. And what are we having? We are going to have T, like small letter T's. So this is our F2, remember. So this is our F2, right? This is our second philo generation. So if you look at these offspring that have been produced, right, we can see that, all right, so these are offspring genotype, all right, these are offspring genotype. So the genotype for offspring are this. Then what will be the phenotype? So the phenotype, so what is the phenotype? All right, so the phenotype for this, we are going to have tall. So this is tall, that is tall, this is tall, and this is short. All right, so what can be our conclusion? Our conclusion is that in this experiment, when we crossed the second generation, we had, all right, we had 3/3 of being what? Being tall, and only 1/4 of that was what? Was short. Right? So meaning that in this generation, right, we usually have, okay, meaning that if these two parents they meet, the chances of them having, so ideally this is a 20, so the chances of this to have a short daughter cell is what? Is 25%. Then 70% it is tall. All right, 75% is tall and that is short. So ladies and gentlemen, that is about this Mendel's experiment and how these genes, or what you are able to see from there. So I don't know if I continue. So in the next video again, I'll give you another example for the last time. Yep.